Picture sitting down to your favorite meal. For me, what immediately comes to mind is thin crust pepperoni pizza. Now imagine what you feel like during those first few bites. There's an explosion of flavor and a really good mood washes over you. For a moment, you think, I could continue to eat this food forever. But as the meal goes on, you begin to feel satisfied. The food may not taste quite as pleasurable as it did in those first couple mouthfuls, and eventually you decide you've had enough and you put down your fork. So you can see that the way in which we taste our food is so closely tied to the decisions we make around eating. With taste, we can determine what the food is. Do we like it? Do we want more of it? So much of the act of eating is governed by this sensory experience. So while you may think that your eating habits are led by your stomach, I'm here to convince you that it's all about what's going on inside your brain. Now, I may be a little bit biased as I am a neuroscientist, and I'm currently working on a collaborative project that focuses on understanding how our bodies, and in particular our brains, oversee the process of eating. Specifically, the goal of my study is to see how brain activity changes when one moves from a state of being hungry, like this mouse about to eat, to a state of being full and satisfied, like this mouse on the right. So the first question was, where do we go looking for these signals inside the brain? Well, we know from past work of many scientists before me that there are several areas that are particularly good candidates of study. These include the taste cortex, which is located at the side of your brain, and the amygdala, which is located at the base of your brain. And while their functions are diverse and endlessly complex, the major role of the taste cortex is to identify the what of the food we eat. So if I'm a mouse eating a piece of food, the taste cortex helps me sense that this food is salty and savory and perhaps identify that it's cheese. While the amygdala, on the other hand, is important for assigning emotions that come along with what we eat. So through the activity of the amygdala, the mouse thinks, wow, I really like this cheese a lot. So think back to what I mentioned earlier. What happens towards the end of the meal when you start to feel full? Those intensely positive feelings may begin to fade a bit and you decide you've had enough. Are these same brain areas that are so involved in taste also involved in that decision? Well, we think so. And a major clue came with the discovery that many of the brain cells inside the taste cortex make a molecule called gastrin-releasing peptide, or GRP. Why was this so exciting to us? Well, we know that GRP was originally discovered in the stomach, where it helps with digestion. So we know that GRP is already involved in eating. And furthermore, previous studies have shown that the activity of GRP can influence how much food you eat. If you inject an animal with GRP prior to feeding, they will eat less. And in humans, GRP dysfunction is present in those with eating disorders. So my job in this project was to listen in on these special cells and to determine how they communicate. So what have I found thus far? Well, we know so far that the brain cells that make GRP largely behave similar to the cells around them. Cells tend to communicate by what you could call a language. They pass chemicals back and forth, each translating a special kind of message. And two brain cells in the taste cortex talk with a common language. What we have found, though, is that it's as if these GRP positive cells are bilingual. They use this common language in most cases, but also they can use GRP to transmit a special kind of message. And our prediction is that this language communicates fullness. What's more is that many of the cells in the taste cortex that make GRP are also in direct communication with those cells in the amygdala, that area giving emotion to taste. And based on my data thus far, I predict that GRP may decrease the activity of the amygdala, thereby decreasing those good feelings as you eat, contributing to the decision to end a meal. So overall, the goal of my work is to add another layer of insight into how we enjoy food. These results add to the framework of understanding why and how we eat what we eat, and may one day contribute to helping those who struggle with this generally essential aspect of life. With that, I'd like to thank my lab and my collaborator, Lindsay Garnacki. Thank you.